Hello, everybody, and welcome to the very last lesson in our DNA, RNA, and protein synthesis unit. So we're talking about protein translation. It's lesson number 5, so last lesson. First things first, we talked about how to make our messages, our secret messages, made out of RNA in the last unit, or sorry, in the last lesson. So now we're going to talk about how we can turn those messages into proteins, right? So how do we do that? Well, when you think of the word translation, what do you think of, right? Like, you probably think of other languages. And in reality, RNA is kind of like its own language. And so ribosomes have to read that language and then turn it into a physical protein as a result. So translation is just the process of turning that messenger RNA into a sequence of amino acids for a protein. mRNA has to enter a ribosome made of rRNA in order for this process to start. So you already have to have your message. So transcription already has to have occurred. So from the previous lesson, in other words, okay? This is a representation of that. So here we have the mRNA strand in this kind of blue text here at the bottom. Here we have our ribosome, this kind of like big, gigantic, bulbous looking thing. And then here we have the amino acids being built using the tRNA. And so in case you forgot, again, we said there are 3 types of RNA, and they're all used in translation. The message, right? That's like our foreman. The ribosome, that's like our construction workers. And the tRNA are literally the tool. So it is handing the ribosome, the tRNA is handing the ribosome the correct amino acid as it's building it. And so keep in mind that once that protein is built, or once the nucleus no longer needs that protein to be built anymore, that secret message, that mRNA, needs to be destroyed. And so it's destroyed by an enzyme called an exosome. And they destroy the message because otherwise the protein would keep being built over and over again. And there are some genetic disorders where the exosomes do not get rid of that secret message. And so proteins just start to build up. Okay, how does translation work? First thing, mRNA has to enter a ribosome. We already kind of said that. Once it enters the ribosome, though, and so again, here's our like broken picket fence looking thing, and that is our RNA. This is our ribosome. Here are our tools being handed to us, right? The ribosome reads the mRNA 3 nucleotides at a time. So it's reading every 3 letters. That's what the ribosome is reading. And that is referred to as a codon. And a code is just a secret message, right? So we literally are translating something. Every 3 nucleotides is a codon. Each codon refers to a specific amino acid, and there are 20 amino acids that this ribosome can kind of use. And the tRNA is what is bringing all of these amino acids to the ribosome. Okay. So how do we know where to start? It's called a start codon. Not very inventive with the name. For ribosomes, start codons are always the sequence AUG. And that codes for one specific amino acid called methionine. It keeps reading, it keeps reading, it keeps reading, it keeps reading until it reaches a stop codon. And stop codons, there are 3 of them. And I'll show you how you can read those in a second. Once that occurs, though, so once we get to the end, we now have a completed protein. Here's a representation of what a protein might look like when it's done. So we're going to practice this and show it. This chart is a codon chart, and it is used in order to read the secret language that ribosomes are using. And so the way you read it is you read it from the center out, and notice, right, U, A, and then here, A is considered a stop codon. So if you ever see UAA, that's telling a ribosome to stop. Also notice, though, G's right there. AUG is also a stop codon. And then UG and A, there's another stop codon here. I just didn't highlight it. Those are the 3 stop codons that tell ribosomes to stop. Let's see if you can find methionine, right? Like that's our start codon. Where's methionine? Methionine is all the way down here. And so what three letters represent the code for methionine? A, U, and then G. That's why you read it from the outside, or sorry, from the inside out. A, U, and then G. That's methionine, okay? And again, I can pick any of these. So what about valine? Val, all right? G, U, and then any letter. G, U, G, G, U, A, G, U, C, G, U, U. All of those code for this amino acid called valine. And that's the way you use this chart. So now that you have your chart at the back of your note packet there, let's actually use it. So given the following mRNA strand, find the amino acid sequence. So remember, we want to read every three, but we always start with AUG. So I see an AUG here, I'm going to underline that, and then I'm going to underline it, and I'm going to underline it, and then I'm going to keep going until I get to a stop codon, but we don't know if any of these are stop codons yet, so let's just stop. Like let's just underline every three. Okay. AUG codes for met. That's methionine, right? Oh, by the way, we're going to be using the three-letter abbreviations because it's going to be way easier than trying to spell the full amino acid. ACG, find that on your chart. It's THR. CCC, that's pro, good old proline. And then UGA is a stop codon. That means that you stop at this point. So we don't need to make anything beyond these three amino acids. That would be it for the secret message. UGA, we stop. You don't write the word stop. And then AAC didn't code for anything because there was a stop codon right before it. All right, number 2. Again, remember, we started AUG. So this first A, meaningless. AUG, AAA, UCA, UAA. Okay, we already know this codes for methionine. So MET. What about AAA? LYS, UCA, SER, and then UAA is a stop codon, so we stop. You might think to yourself, oh, this is really easy, right? Like, we only have 3 amino acids. These can be very, very long. Proteins can have hundreds of amino acids. So this RNA message could be going on for quite some time. And that is the end for our secret message. So again, if you have any questions, please let me know. But that's our last lesson, so fill in the notes as you go.